Awesome. Yeah, can you hear me? Awesome. Okay. Hi, I'm Keller. I'm a research associate at Parallel Square Technology Institute on the immunology team. And I have the honor of introducing our first speaker today. Dr. Galit Lahav received her PhD in 2001 from the Technion, Israel Institute of Technology. In 2003, she completed her postdoctoral fellowship at the Weizmann Institute of Science in Israel. She then spent a year at Harvard's Bauer Center for Genomics Research, and in 2004 joined the Department of Systems Biology. In 2018, Dr. Lahav became the chair of the department. Dr. Lahav pioneered computational and quantitative experimental approaches to studying the fate and behavior of human cells at the single cell level. Her work has yielded critical insights into the function and behavior of tumor suppressing mechanisms and their role in cellular destiny. She has been recognized for excellence in both teaching and research through several awards, including the Smith Family Award, Vilcek Prize for Creative Promise, and Excellence in Teaching and Mentoring Awards. Lahav is now leading a department that works across traditional disciplinary boundaries to create new formulas, conceptual and mathematical models that enable moving from observing biology to predicting and engineering it. Please welcome me in welcoming, please join me in welcoming Dr. Lahav to the stage.
Thank you, Keller, and thank you for the invitation. It's a pleasure to be opening this very exciting meeting, and a meeting that is so easy to get to from, as you said, across the road. I'm very excited to share with you a recent story from the lab, and a story that I think the next steps of this story will really benefit from what we're all going to hear here in this meeting. But I will start by providing a little bit of background on the main goals of the lab and some of the critical information and the things that we learned in the last 2 decades. And do you have a clicker? There you go. Thank you. All right, so I think we will all agree that this clicker doesn't work. It's okay.
Oh, it's because I don't know why it's not working.
Is there another way to advance the slide? I will start just talking while we figure this out. So what I was about to say is that we all agree that in the last 2 decades, it became clear that cellular information, and I mean both information within the cell, but also information outside the cell, can be transferred through what we call protein dynamics. And by the word dynamics, I mean anything that we can measure over time. It can be concentration of protein, localization, modification, but it's really the idea about the shape of the curve, how these levels change when you look at them over time. And when you start to look at these measurements, you realize that it's actually very rich, and you can look at parameters such as frequency, if there's oscillations, amplitude, area under the curve, duration, and the computer is restarting. We are quite obsessed in the lab in trying to understand how information is encoded in these parameters and how these parameters are decoded by cells to trigger the right output in response to different external and internal inputs. And we've been using this umbrella to study a lot of processes in biology, cell cycle regulation, cell size, DNA damage and repair. What I will show you today is one of the main pathway that we've been studying, which is the p53 signaling pathway. So what do you need to know about p53? The p53 biology is huge, but let's try to summarize it and make it very simple. p53 is a tumor suppressor. It is the gene that is mostly mutated in cancer, and it responds to stress from the environment. And so when cells encounter cellular stress, or in our case, we mainly focus on DNA damage, p53 levels go up, and then it activates many target genes. It's a transcription factor. And what's quite interesting about p53, it has like these bipolar personality or dual personality. On one hand, it can activate programs that will allow the cell to cope with the stress, to cope with the damage, and resume growth. So it triggers more like survival. On the other hand, it activates genes that participate in terminal phase, such as senescence and apoptosis. Okay, and the notion is that if the damage is too big, it's too difficult to cope with, it's better for this cell just to die or to arrest forever, and in order not to continue to grow, because when p53 is mutated, these damaged cells grow and can develop into tumors. Okay. So a lot of effort has gone in the entire p53 community to understand how this one single transcription factor chooses between these different outcomes, right? Sometimes it activates survival and repair, sometimes arrest, sometimes apoptosis, sometimes senescence, and so on. And a lot of beautiful studies suggesting it's cofactors that bind p53 or the level of p53. And when we entered the field, we were wondering whether dynamics matter, right? Whether different inputs trigger different dynamics of p53 and different dynamics of p53 leads to different outcomes. There we are. Okay, so here's our p53 responding to stress and activating different downstream targets. And the question that we had was...
Right now. Nope. Sorry, like...
The question we had is, do cells use modulation of p53 dynamics to achieve specificity in this network, so that one dynamics will trigger one outcome and another dynamic another outcome? And so what you need to know about when studying p53 dynamics is that the regulation on the protein level is through the stability of the protein, okay? So there's no, in response to DNA damage, it's not changes in the mRNA of p53. It is, it is changes in the protein stability. So p53 activate the transcription of a target gene called MDM2. MDM2 is an E3 ubiquitin ligase. It binds to p53, ubiquitinate it, and send it to degradation, right? So we get this very classical negative feedback loop with two different arms. There is a transcription arm, which is a slow rate response, and protein-protein arm, which is a fast rate response. And even with this simple negative feedback loop, depending on the parameters, you can get different behaviors for p53. You can get oscillations, you can get just single pulse, you can get accumulations, and so on. Okay, so it gives you an array of dynamics that may be used for p53 function. And one of the things that we discovered many years ago is that in response to gamma radiation that leads to double-strand breaks in the DNA, p53 show this series of beautiful pulses in cells. So if we look at the individual cell level, we see oscillations in p53 of a period of about 5 hours, 5.5 hours. When you average all them in the population, it seems like they're damped because with time cells lose synchrony. But within each individual cells, we get these pulses of p53. And we also saw that we don't always get oscillations. It depends on the input, right? So gamma radiation leads to these oscillations, but if we give different kind of damage through UV, we get one single pulse. If we give a drug like cisplatin, we get accumulation and so on. So we tested different ways to activate p53. We got different dynamical behaviors, hinting that maybe dynamics is used here in order to transfer information. And this kept us busy for many years, understanding the molecular mechanisms of all these different dynamics. But the real question that we have is, do cells care, right? Do cells care if p53 oscillate or doesn't oscillate? And in a series of studies, we looked at perturbation of p53 dynamics and look at the impact on target genes of p53. And here is one example. If we just irradiate cells and we get these oscillations, this allows recovery and growth. But if we use a small molecule that inhibit MDM2 and sustain p53 levels, then it pushes cells towards senescence, right? So that was the first example we showed that just by tweaking protein dynamics, just that, very accurately, right, we can activate different target genes genes, and we can push cells toward different fates. And a couple years ago, we also took it in vivo and measured p53 dynamics in different tissues and found at this point correlation that the more sensitive tissues showed sustained p53, the more resistant tissues showed oscillations. Again, so oscillations are more related with recovery and growth, and the sustained more with permanent cell cycle arrest. And we also used a xenograph we measured p53 exactly as we did in cell line. We found the exact same thing. If we use a small molecule to change p53 dynamics from oscillatory to sustain, right, then we actually lead to a better control and shrinkage of tumors. So basically, in all these studies, we measured total p53, right? Because we use a reporter, we take p53, we fuse it to a fluorescent protein, and we just follow it over time, right? So it's total p53. But we know for many years that p53 can be modified a lot. And a lot of acetylation and simulation and phosphorylation and any type of modification that you can think of, p53 has it, okay? And so a few years ago, we started to wonder whether different pulses of p53 have different modifications. So if we look at the first pulse, maybe it's modified in a few sites, and then if we look at the second and the third, maybe it's modified in other areas. So even though total p53 look the same, these pulses have the same amplitude, the same period, the species of p53 in terms of modification, does it change between pulses? And the best way to do that is through mass spectrometry, right? And in the last, 15 years, at least more than 10, we've been collaborating with many across the US to try and use mass spec to probe modifications on p53 for reasons that I don't fully understand. p53 is one of the most difficult proteins to do mass spec on. I'm happy to talk with each one of you during breaks and coffees and tell you about all the different things we tried and what didn't work. But at some point, one of our collaborators from Northwestern, Neil Keller, I know we have another talk from this group a little later today, suggested that we do something different, which is a system that he was working on at the time, which is top-down mass spec, which is basically you don't digest the protein, you take the entire protein and you run it through a mass spec, and you can identify things that we call mod form, different modification forms of p53. So we don't know where the modifications are, but we can tell how many different species they are and what is their abundance, what's the distribution of these. And so we said, you know what, that's good enough. Let's start with that, right? And we can compare the first and second pulse of p53. If the mod forms are exactly the same, then we're done. That means that they're similar and we don't need to dive deeper. But if they're different, then we'll try to find new ways to understand what these modifications are. So this is how data from mod form look. Here, this is the least modified protein, and the green go here is the more modified, and you get distributions of species in different modifications. And so we did this for p53. We took samples from the peak of the first pulse and the second pulse. And this is the spectrum that we get, first pulse and second pulse. Again, here is the least modified p53, and here is the most modified p53. And when you compare both of these, they look pretty similar. But if you look carefully, you see that there are a few places in which there is a species here of p53 that doesn't exist in the second pulse. And again, here's another example. Exists in the first pulse and not in the second pulse. So that was enough to motivate us in order to try and identify which potential modifications exist in the first p53 pulse and not in the second. And then we went into the good old Western, and we used all the possible existing antibodies to find which modification may change between the first and second pulse. Here is an example of few that we tested. We look only at first and second pulse of p53. And many of them don't change, they look the same, or they're just not there. But we realized that two of them, acetylation at 373 and 382, appear in the first pulse and disappear in the second pulse, right? Even though we load similar levels of p53. And if we look at them over time, we see the same behavior, right? This is total p53. We see the first pulse and the second pulse. Remember, here it seems a little damp because cells lose synchrony. But when you look at the acetylation, it exists on the first pulse, disappear on the second pulse, okay? So this is one modification in which the species of p53 is different between first and second pulse. What could lead to this difference? Oh, here, if we quantify it, we see this is total p53, and this is acetylated p53. And so with that, let's see if we can actually change this by using small molecules and inhibitors to understand the function, the potential function of these modifications. And the idea was to take the same experiment in which we have total p53 having two pulses that are the same, and acetylated having one pulse and a smaller pulse, and then to add a deacetylase inhibitor between the two pulses, right? And see if we can enhance the acetylation on the second pulse. And the answer was yes, with one particular inhibitor. We saw that when we use an HDAC inhibitor, then on the second pulse, then we can enhance p53 acetylation on the second pulse, okay? So telling us that during the second pulse, there are deacetylases that remove this acetylation from p53, and this is why we see the difference between the first and second pulse. So what we can do now, we can use this similar setting and look at target genes of p53 and see whether they care at all, if it's acetylated or not acetylated. So we started with just a set of what we call canonical target genes and compare their levels in the same setting I showed you during the second pulse, right, without HDAC inhibitor and with HDAC inhibitor. And you can see that some target genes, like this is P21, a cell cycle arrest protein, GAD45, that's a DNA repair protein, another DNA repair. Some of them don't really care, right? The induction is pretty similar, but there is a group of genes that really like P53 to be acetylated, because when we add the HDAC inhibitor, that leads to much more acetylated p53, we see a strong induction in the level of these genes. And this is an apoptotic gene, PUMA, and these are two genes participate in senescence. Of course, HDAC inhibitor does a ton of things to cells. We could show that if we knock down p53, we don't see that difference, but this means it's p53 dependent. It doesn't mean it's the acetylated dependent. So in order to really prove that, we had to go and mutated these two sites and see if we see differences in target genes of p53. So we created mutation in one of the residue that has chain 373. In the second one, we also, as a control, created a mutation in another acetylated site that did not change, 381, and we created the 2KR that has both of these. And just in order to squeeze everything in short time, I will just show you the final results that we got for the 2KR and how it impacts target genes of p53. So basically what we did, we had created two cell lines. One is expressing the regular wild-type p53, and one is the mutant that cannot be acetylated on these two sites. And we did RNA-seq and collected samples for the first 3 hours after radiation and compared, okay, globally, all target genes of p53 and basically all mRNA between these two. And I would just say what we expected to find and then what we find. So I was under the impression that what we'll see is either genes that don't care if it's acetylated, like what we saw before, and many genes that will love it when it's acetylated, that will prefer it's acetylated. But the picture was a little bit different. And let me show you this piece of data. So what we see here, this is the expression level of a target gene if p53 is mutated. And this is the expression of a target gene if p53 is wild type. Okay, the wild type can be acetylated, the mutant cannot be acetylated. Everything in gray here are genes that don't care, right? They are induced similarly, whether it's acetylated or not, right? Whether it's wild type or mutant. Everything in red here, these are genes that have preference for the wild type, meaning that p53 can be acetylated. These are the ones that we expected to find, right? That they prefer acetylated p53. But then there was this group here of these blue genes that actually prefer non-acetylated p53, okay? That they are induced much better when we put the mutant that cannot be acetylated during the first pulse, all right? So we realize, and as you can see, this is really gradual, right? It's not like a complete separation. You can see that there are genes that care a lot if it's acetylated, and genes that care a little bit, and those that don't care at all. So, the emerging picture from everything I told you is that there is an interplay between the dynamics of p53 and modification in order to temporarily control the different programs downstream of p53, okay? There is a gradual, if you look at p53 target genes, some of the genes prefer acetylated, some don't care, some prefer deacetylated. And so in the first pulse, when p53 is acetylated, it pushes more the programs that prefer acetylated p53. And then in the not acetylated, in the second pulse, it activates a different program that prefer the non-acetylated p53, okay? And in a way, this study has been very satisfying because for many, many years, we're asking ourselves why p53 oscillate, like at all. And this suggests, if you think about a goal for a cell to temporally order something, if the gene doesn't oscillate, if the protein doesn't oscillate, then you are left with protein from the first pulse that may have modification from that pulse. But if it's oscillating, then you get rid of all the protein that was there and you're expressing a new fresh protein that can be modified in different areas. What we would love to do here is to take these modifications in the single cell level and to combine our live imaging with measuring modifications with single cell proteomics. So I'm very excited about what I'm going to learn from this conference here. So this is what I wanted to tell you today. These are the people in the lab and our main collaborators. The postdoc who was leading the work on p53 modification is Dan Liu, and this is in collaboration, as I said, with Neil Keller from Northwestern, Jeremy Guanadena at HMS. And thank you so much for your time. I'm happy to take questions. I love questions, so happy to take them. The timing of each pulse of p53 is about 5.5 hours in human cells.
So, do you also see the modifications of p53 within the same pulse, like how it changed? Because as you said, it's a 5 hours, which is very long time for a protein to change the dynamics.
Right.
And also, when you take the samples from different pulses, what is the criteria? Are you taking exactly the same time for the both pulses to compare, because apple to apple comparison? So I'm just curious, like.
Yeah. So the total p53 we can measure at the single cell level through dynamics, right? So we sample it every 10 minutes, right? So we get a really high temporal resolution. When we look at the modifications with mass spec or with Western, we look every hour or every half hour. So basically the temporal resolution is different. And when we compare them, we take the peak of the first pulse and we take the peak of the second pulse, right? So we take when the protein is highest. But remember that when you do it at the population, the first pulse is the most synchronized. The second pulse is already a little less synchronized. So it's probably not going to be the peak of all cells, right? On average, it's the peak, but probably some cells are still on the way up and some already starting to go down.
First and second pulse, use about how many pulses you have been quantified with PTM.
We normalize this to total p53 to make sure that because the levels are different, that's not the reason that we see less acetylation. Yeah. Yes. Hello, Katie.
Hello. Thank you so much, Galit. It was a really beautiful talk. I'm wondering for your acetylation modification, do you think that you're actually getting differences in binding to those target genes, or is this a more indirect effect?
Oh, I love this question. So I think we know the answer to that because a few years ago, we actually looked at p53 binding to its target genes, to DNA, during the first and second pulse, and it was exactly the same. Okay, so it's not binding. We also see p53 bonds in tons of areas in the genome, but the activation is much, much smaller. So I think it impacts the ability to bind some cofactors or that impact the transcription itself and not the binding.
Do you think these modifications are affecting the transcription point more, or is it affecting the stability and interactions of MDM2? And it's actually, kind of affecting the pulses, like the acetylation may encourage degradation or not?
Yeah, that's a great question. I think because the mutant still oscillates, I don't think it impacts the stability or the feedback with MDM2. And it is known that the stability with an MDM2 is mostly affected by phosphorylation on p53. So I think these are more impacting the ability to transcribe DNA, mRNA. I cannot hear you.
Very beautiful work. I
Thank
have
you.
a very general questions. There are a lot of mutations on p53, and some are functioning to impact the patient's outcome. But in terms of the time scale, the pause may affect the binding of or recruiting of other partner or the protein modification, which is faster. The change of the modification or the interacting partner, which may come first, and how will the mutation also change the dynamics?
Yeah, so you're asking about the mutation that we use, the acetylation, or mutations in cancer? Both, okay. So as I just said, the modification on the acetylation, the inability to acetylate, doesn't change the dynamics. They still oscillate. Hotspots in cancer are usually mutations in the DNA binding domain. So you get a protein that cannot activate target genes. It cannot activate MDM2. So it totally disrupts the oscillations, right? You get completely different dynamics. And in terms of the speed, so the induction in the level of p53 is relatively quick. And it's a little hard to distinguish between the speed of modification in protein, because first you need to have a protein in order to modify it, right? What we do see in the first pulse, it basically mimics each other. So the same kinetics that we see from the protein, we would see for the modified protein.
Hey, wonderful
Hi.
talk. I'm Hong from Chinese Academy of Medical Science. So I have two questions. So the first is a technical question is when you do the opt-down aspect, you see the two peaks which are different, and then you did Western to see the acetylation, does it match? That's the first question. And the second question is you measured the first pulse and the second pulse. How about the following ones? What do you think is every pulse mean different biology or you just
Yeah, no, these are they're great questions. So we can't tell if they match because the mod form, the top-down, doesn't tell us where the modifications are. Neil's lab group are working on that, and I think we'll get to a point that we'll be able to figure this out. But for now, we just know that from the top down they're different. We don't know where. In terms of the subsequent pulses, that is a super interesting question, whether they look like the first, the second, something different. Technically, we're a little stuck in our ability to do it until we can do single cell proteomics and measure these modifications at the single cell in a way that is not antibody based, because these antibodies don't work well for immunofluorescence. So please develop a method so we will be able to answer this very exciting question.
This is Patrick from TGen. So I think you already answered the question, but you don't really know where it's happening, right, on the DNA. So I was wondering if you did open, any kind of ATAC-seq and looked at protein states between the pulses. Would you imagine it changing a lot? That's what I would suspect.
Yeah, we haven't done ATAC, we haven't looked at that. I think looking at chromatin state and see how it changes will be fascinating. So that's certainly something we would love to do. I'm at Harvard, we can't hire anybody, right? So we're a little stuck in our ability to do any of these projects, but that is on the list. This is like the next most obvious exciting things. I agree. Yeah, maybe someone wants to collaborate. Yeah.
I wonder how the oscillations happen. So is this transcriptionally driven, or this is purely on protein, like the protein is changing, but the mRNA is not changing? And maybe also how common is this mechanism? So the oscillation, is it a p53 specific thing, or you can think of many other regulators behave the same way?
Yeah, so on p53, the oscillations are at the protein level, not the transcription. mRNA is flat, it doesn't change. It's really just the stability of p53 through MDM2. And you're absolutely right, p53 is not the only one that oscillate. The upstream kinases that activated, like phospho-ATM, phospho-CHEK2, they oscillate as well because there is a loop from p53 back to them. So there are a lot of things that are oscillating, right? It's a little hard to say what is the cause and what is the result, but a lot of protein at the DNA damage response oscillate. We actually did a mass spec to look at everything every hour, and we see a lot of DNA repair protein that oscillates, phospho protein that oscillates. So this is just probably one of them.
Nikolai Slav from Northeastern and PTI. Wonderful talk, Galit. Very much enjoyed it.
Thank you.
And one expectation that I have based on it is that if you're able to measure nascent transcripts, you'll be able to see that the first pulse is associated with higher increase of those genes whose transcription was more induced without acetylation, and conversely, the second pulse would be associated with more nascent transcripts corresponding to the different mod form. Can you measure this? Is this also a way that you can use to identify differences in transcripts induced in more pulses?
Absolutely. Yeah, I think that is possible. We have, again, from a few years ago, another study that we look at mRNA and nascent transcript, but we haven't connected these two, but I think that is very feasible. Yes. Yeah.
Thank you.
Thank you.
Any other questions? I think there might be one online.
I think we have that.
Yes. As we move forward with more and more data, can we turn it into music? The trained ear can detect a wrong note.
As we move forward with more and more data, can we turn it into music? The trained ear can detect a wrong note. We could. I think if we want to train oscillations into music, I'm up for it. I love music. So yeah, reach out, whoever wrote that question, and we'll turn. We have a lot of movies that we collect, and I always feel like we need to have some soundtrack to put it in the background. So that could be a way to do it. All right, thank you.